Alright guys, welcome to Target PEBC Prep. My name is Mohit Koladia. Today we'll be starting a discussion on ion channel receptors. So this is another important type of receptor that you need to know. Sorry. So basically in this there are three types of ion channel receptors mainly. First one is the ligand operated. So this is the most important one and we'll be discussing with respect to the drug mechanisms. Okay, so the ligand operated one is the one that works when a ligand binds to the ion channel. It shows its mechanism. Another one is the voltage operated. So the voltage operated ones basically works with the fluctuation in the voltage across the membrane. So this channel has a closed and open mechanism that works via voltage. And the third one is the mechanically operated. Okay, so this channel works basically with differences in the mechanical pressures, or whenever there is an exertion of mechanical pressure on the receptor, it works. So the most important one we need to remember is the ligand operated one, because most drugs that work on the ion channels, they work through the ligand operated mechanism. Now there are two types of effects that you will see usually with ligand operated ones. The first one is the excitatory transmission. Okay, so in excitatory transmission, and the second one is the inhibitory transmission. Okay, now in the excitatory transmission, the examples include nicotinergic receptors. Second is the 5-HT3 or serotonin receptors, and the third one here is glutaminergic or glutamate receptors, or it's also known as NMDA and methyl D-aspartate receptors. Okay, in the inhibitory you have to remember two, that is the GABA and the glycine. So let's discuss each of this in detail. Okay, let's first start with the excitatory transmission. So just remember, see, all the receptors or molecules that have excitatory effect would always work in this way. Okay, it could be on a cell or it could be on a neuron. Okay, so what happens in an excitatory transmission is basically, say for example you have a neuron here, pardon me for my drawing, and you have a neuron, another neuron here, okay. So say this neuron, it releases neurotransmitter, and this neurotransmitter then acts on the ion channel on the surface of this receptor. Okay, so the basic mechanism in excitatory is, when this neurotransmitter binds here, it basically opens the channel which allows the entry of cations. Okay, it could be calcium or it could be sodium or any other cation. So basically it's cation loading. So the cation enters into the neuron and this leads to the depolarization. Okay, just remember this term, this is important term for pharmacology. So there is a depolarization and the cells, they get excited. So in the like when the neurotransmitter is not bound, the resting membrane potential is basically, say, minus 90 millivolt, okay? But when the cation loading is done, this negativity is decreased and the threshold is reached, which is maybe 20 millivolt, okay, for example, just so this is the basic mechanism of excitatory transmission: cation loading and depolarization, and this leads to the rapid firing of the neurons, okay? Right? So now this, as I told you, the examples are nicotinergic. So when it's nicotinergic receptors, what would happen is this neurotransmitter would be ACH, acetylcholine, which binds to the channels. These channels would be nicotinergic receptors on nicotinergic receptors, okay? And another exogenous molecule that mimics this action is nicotine. So these receptors are called nicotinergic receptors, okay? Now let's discuss the another one. So what is the another example that I told you? Come on, yes. So the another example for this should be 5-HT3. So another example is also 5-HT3. So when it's 5-HT3, this neurotransmitter would be serotonin or 5-HT3, which binds here and leads to rapid firing. Now the important thing to know here is the antagonist of these receptors have a very useful pharmacological effects. One is the anti-emetic, the other is antipsychotic, and the third is anxiolytic. So 5-HT3 antagonist, you have to remember which blocks the binding of 5-HT3 here. So if you block, the firing is slowed down or there is an inhibition. So those antagonists work as anti-emetic, antipsychotic or anxiolytic, okay? Alright, now the other one that you need to know is the other important one that you need to remember here is the glutaminergic, or okay, for glutaminergic I need to explain you because it has slight different, not much, it works in the same way, but it has a slightly different mechanism, okay? It does the same thing, but the excitatory part comes from slightly different mechanism. So what happens is, say this is the ion channel receptor, okay? So on its resting stage it has magnesium bound to its surface. Now when the neurotransmitter comes and binds to this receptor, that is glutaminergic, okay, glutamate receptors. So when the neurotransmitter binds here, along with this the receptor has, so this is cell surface, okay, so along with this the receptor has an internal voltage operated mechanism. So this works in two ways. One is the receptor binding, that is the receptor is bound by the ligand and the ligand bound leads to the voltage fluctuation, which also helps, and this voltage fluctuation leads to the opening of this, and once this opens the Mg+2 is released, okay? So in that case the channel would look something like this. So the agonist is bound here and the magnesium is gone, and which allows the entry of other cations. Okay, so the voltage plays a role here from the inside, as well as the agonist binding helps. Now the important antagonist here is phencyclidine. Just remember this, phencyclidine blocks this binding of the agonist. Okay, the another one is very funny name, diazepam. Okay, so the effects of this antagonist is basically if you block this receptor, you develop hallucination. So these are hallucinogenic agents. Okay, antagonists to glutamate receptors are hallucinogenic agents. Okay, just remember this. And this glutamate receptor are mostly located in hippocampal area. So in hippocampal area, if its location is in hippocampal area, it basically plays role in memory functions. Okay, so you are remembering the things right now because your glutamate receptors are properly functioning. So you remember what you study because of hippocampal area memory function by glutamate receptors. Perfect. Now let's go to the inhibitory transmitters. So again, these are important to know. Inhibitory transmission. Okay, so here you have to study, first one is the, okay, let's first discuss the basic way how it works. So the first important one, how inhibition work is, say from this neuron a neurotransmitter is released, it again binds to the surface of another neuron that has ion channels. Now there are two mechanisms here. The neurotransmitter binds here, so binding to the neurotransmitter leads to K plus efflux. So the potassium ion gets efflux. Okay, so there are two mechanisms. One is the potassium efflux leads to hyperpolarization of the cell. Because K plus is moving out, you are losing the positivity from the cell, so your cells is becoming more electronegative. So here if it was minus 90, now it's going to minus 110 or 20. Okay, so it's becoming more hyperpolarized. And the another mechanism is this neurotransmitter, they binds to the ion channels and leads to chloride influx. Okay, so there could be chloride influx, and this is also leading to hyperpolarization because your cell is becoming more and more electronegative. So this hyperpolarization effect leads to inhibition. So most of the drugs that have inhibitory transmission effect will basically work through either K plus efflux or increasing the Cl minus influx. Good. Okay, now let's understand the important ones. The GABA. You all might be aware of very important agents that work through GABA, right? Okay, so the first one is the GABA. Let's do it side by side because it's easy to remember here. And the other one is glycine. Now both work through the same way, that it increases the chloride influx and leads to hyperpolarization. Both works through same way. Only difference is this is located throughout CNS. Okay, GABA receptors are throughout CNS, while this is only in spinal cord. Now for GABA, you basically have two receptors, GABA A and GABA B. So GABA B we won't be discussing because it's operated through G protein coupled. Okay, it works through G protein coupled, and GABA A is basically ligand operated ion channels. So what it does is GABA A binds to the channel, GABA binding to this GABA A receptor leads to chloride influx and hyperpolarization. And hence it leads to inhibition. Okay, same way glycine also works in the same way. So just mechanism is same, but you have to remember GABA A because most benzodiazepines, barbiturates and steroid metabolites, they are GABA A agonists. Okay, so your GABA A has benzodiazepines, barbiturates and many steroid metabolites. Okay, so these are agonists. Now the benzodiazepine, they actually increases the frequency of chloride channels. So it increases the frequency of opening of chloride channels, so it blinks again and again. Okay, open close, open close, open close. So it increases the frequency of opening of chloride channel and leads to inhibition of firing. And barbiturates, it prolongs the duration of opening. So the only difference is frequency in benzodiazepine while the duration of chloride channel opening in barbiturates. Okay, so this also leads to inhibition, but barbiturate prolonged duration of opening would be dangerous, very dangerous. And many steroid metabolites also work through opening of chloride channels. Okay, so these are the agonists of GABA A. Now let's discuss slightly. Okay, let me tell you what are the antagonists. Okay, antagonist, maybe you can remember this, that is picrotoxin. You have a funny name, bicuculin. Again, one more is penicillins. Okay, all three are GABA antagonists. Alright. Thank you for watching my video. Please press like or subscribe and share with your friends. Thank you very much.